Hi, my name is An. I'm a fourth-year PhD student at the Mayo Clinic Graduate School of Biomedical Sciences in the Biochemistry and Molecular Biology track. I am a recipient for the PhRMA Foundation Predoctoral Fellowship Award in Drug Discovery. I became interested in science from my first chemistry class. I think it was really cool that, science gave us a way to visualize and explains processes that happen in the natural world that we don't necessarily see by eyes. And everything just makes sense, right? And so research is a way to gain new knowledge and open our eyes to new things that haven't been explained or discovered yet in science. And I really enjoy that process. So my project aims to find a new way to prevent a very toxic side effect that happens in a lot of cancer patients undergoing chemotherapy for their treatment. Common chemotherapy uses a family of compounds called anthracyclines that target a small family of protein in our body. These compounds are very effective, but they kind of act as a double-edged sword where they target two different versions. You can think of it as sister proteins inside our body that carry out the same function, and they're very similar to each other. Targeting one version can very effectively kill cancer cell, but then targeting version number 2 leads to a lot of the heart damage that we see in patients who survive cancer therapy. I'm trained as a molecular and structural biologist, so I'm very much interested in how structure impact function inside in these proteins. And structure now has given us the ability to discover new binding sites. We identified a new binding site for inhibitors, which is like a new lock that has some slight difference between the two versions. So our research basically aims to identify new keys that can very specifically fit into one lock, molecules that we can use to target one version of the protein over the other and prevent the activity of the versions that causes cardiac or heart damage in patients. Whereas we still leave the other version fully functional, and it can be then targeted for cancer therapy. When I started my PhD, this wasn't my original thesis project. So this project just really happened out of a conference where my PI went to meet with new scientists in the field and started this collaboration. And I was the one who, spending half of my time working on it and half of my time working on my main thesis project, which aimed to study mechanism and function of these proteins in our body. And slowly it has really turned into a big drug discovery project, and I didn't realize that I would enjoy doing this work so much, because when I started, I never thought that I would be doing drug development or discovering new compounds and new therapy. So this has been a very important turning point moment for me, because now I'm seeing a completely new trajectory in my careers that I can aim forward, and I'm still keeping a lot of my enthusiasm and curiosity for studying mechanism and function of proteins. We're very driven by our curiosity, and we want to find the answer. We want to find a logical explanation for things and figure out how things work and why it's behaving this way. And a lot of it is also ties to the fact that I'm very stubborn. So, if things fail, I feel like, maybe I should try again, or maybe I should approach it from a different perspective. So once I finish my PhD, I naturally probably postdoc, but I would really much like to see myself working as a research scientist or in either, academia, maybe more of a research institution, or R&D in the industry.